Well, thank you to all for being here. I'm gonna, share with you this amazing project about on-site nanopore sequencing on Colombian Pacific Coast to reveal the mangrove-associated soils microbial diversity. So in the first place, Colombia is located in northern South America. It's known for its wide biodiversity. The project took place in Bahia Malaga, a small port on the Pacific coast with no airports, no connecting roads, and it was a significant challenge because we had no access to electricity, no running water, no any connectivity services. And the bay relies on tourism as their economic driver, and well, this zone is also very rich in mangroves. They are really important for the coastal protection, for sequestering carbon, and for all the diversity in general in the area. It's also important because this is the perfect habitat for piangua, a clam-like mollusk that is really important for the local economy, especially benefiting women. This project is part of a much bigger study that aims to study piangua through genomic population studies and to assess its microbiome. Now, diving into the second segment of our talk, the field sequencing. This is a really interesting part. All of the devices were taken on styrofoam boxes from Bogota to Bahia Malaga. We had to travel from plane, then rough roads, a boat, and the enzymes and all of that were stored in fridges to try to maintain their temperature. For the technical aspects of our study, all of the devices requiring electricity were connected to solar panels. The DNA extraction was made there on field from samples that we took from multiple points along the mangrove bay. And the PCR amplification was made there using a portable thermal cycler. And we amplified the 16S gene for bacterial identification. It's really interesting that this step of library preparation was was made there on the field, and the community participate actively in this stage, and it was really good to see them engaging with laboratory equipment for the first time in their lives. The sequencing was made using this portable Minion sequencer with the complemented R9 kits, and then data analysis was made after that on the lab in Bogota. As you can see here in the video, the sequencing yield was really good. The flow cell retained over 1,200 pores even after all of the travel, and we obtained really good results. Now for the last segment of the talk. As you can see here, the rarefaction curve shows the effectiveness of the sequencing. As the library size increases, the curve steadily ascended. This suggests that most of the microbial diversity was captured by the sequencing process. This can tell us that our study had achieved a comprehensive understanding of the microbial landscape within the samples. Here, for the relative abundance, we can see two distinct approaches of analysis, one made through epitome on the right. Yeah, well, it's that one. It's the one on that side. Sorry. And an independent build pipeline. As you can see, in the independent build pipeline, we got a higher quantity of bacterial phyla, and epitome only depicts the most representative ones. This doesn't undermine the effectiveness of epitome. Epitome is still a great platform. It has been really updated. Now we have more accurate results, but it's always subjected to improvement. For our conclusions, our project in Bahia Malaga will demonstrate the viability of taking this technology and the laboratory to the field, even to challenging conditions, as I told you before. Epitome workflows have been optimized from what you had a year ago. Now it's a complete whole different world. They're really good. They do reliable analysis, fast analysis, and as I told you before, it's always good for improvement, but it's really good. And the integration of the portable equipment like Minayan contributes to understand the microbial diversity, but it is also really good for another future projects to be made with this technology in the field. And thank you so much, and I'll be happy to take your questions. And thank you so much to my colleagues, especially Mariana Restrepo, Marcela, and Silvia Restrepo.